Hello, my name is Sophia Hayes and I'm the head of the Molecular Spectroscopy Lab in the Chemistry Department of the University of Cyprus. My lab is primarily a vibrational spectroscopic group with resonance Raman spectroscopy as our main research tool. Our mission is to provide a fundamental and molecular level understanding of how chemical systems work, employing different structural probes in order to generate design rules for better optoelectronic devices, pharmaceuticals, and new bioelectronic devices. Our spectroscopic tool, resonance Raman spectroscopy, allows us to investigate a whole gamut of chemical systems. Our research spans different fields from organic electronics to biophysics and the intersection of the two bioelectronics. Starting from the organic electronics field, we're interested in generating structure-property relationships in order to understand the effect of structure on the optoelectronic behavior of these materials, and hence identify their best use for applications such as photovoltaics, light-emitting diodes, transistors, photodetectors, and so on. We do this by studying the molecular conformation of conjugated polymers under different conditions such as chemical substitution, physical blending, thin film processing, temperature, and doping, that is oxidation or reduction conditions. As a molecular spectroscopy lab, we don't work in isolation, but we do try to bring a holistic approach to organic electronics. We combine the molecular structure information obtained in our lab with macromolecular structural information, for example, from imaging methods such as AFM or the X-ray diffraction methods such as GWAX, along with ultrafast spectroscopic methods that provide insights about the photophysics of our systems under study in order to get a rounded knowledge of our chemical system. In the biophysics field, we've been involved in different projects, an example of which I'm showing here. Essentially, in this field, we're interested in how small molecules can interact with biomolecules, whether these biomolecules could be proteins or DNA. In particular, this example I'm showing here had to do with the identification of the interactions that extend between small molecules and the G-quadruplex structure of DNA, which was used as a pharmaceutical target for anti-cancer drugs. Resonance Raman spectroscopy can give us specific information about these interactions between the small molecule and the DNA, and this can help us understand these systems better. The bioelectronics field is at the interface between organic electronics and biophysics. One of the goals of this field is to develop interfaces that sense or stimulate biological systems using materials that have good optoelectronic properties, such as conjugated polymers, which is what we use in the organic electronics field. Within bioelectronics, we have been involved in a number of projects. For example, the development of a biosensor working as a fluorescent switch that detects interesting DNA structures like the G-quadruplex DNA structures with potential in detecting them in cancerous tissues. Other conjugated polymers can be used as sensors for DNA damage, such as this cationic polythiophene we had investigated. Conversely, the biomolecular DNA can impart new properties to the polymer by acting as a template for its conformation. Within the framework of this project, we'll work towards understanding the photophysical properties of this complex chemical system using a variety of spectroscopic methods, assessing the influence of each partner on the photophysics of the other. This helped us understand, for example, whether this novel structure could be used in new optoelectronic applications. To sum up, at the Molecular Spectroscopy Lab, we use resonance Raman spectroscopy as our primary research tool to help us gain a better understanding of our chemical system. Then, in combination with other spectroscopic methods, we can build structure-function relationships that can lead to improved optoelectronic devices, for example, and new technological applications. Understanding the interactions between very small molecules or macromolecules and different biomolecules, we can design new drugs or new diagnostic and or functional devices for the early detection of disease or health monitoring. With this, I'd like to thank you for your attention.